Uh, Hello, everyone. So today I'm going to talk about inferring metapopulation movements from kinship estimates. But before I start, I'd like to thank my co-authors on this research, which is Dr. Mark Lethbridge at Flinders, Dr. Ben Allen at USQ, and Dr. Rose Andrew at UNE for their contribution to this work and also the broader project. So for context, there we go. So for context, properties in central Western Queensland and actually across Australia have erected exclusion fences to help the property managers better control their pest species. And when done right, these actually work really well. As you can see here, there's no kangaroos on the other side of that fence. But obviously, if there's other wildlife species in the system that are bigger than the mesh holes and that can't fly, they're also going to be affected. And they could be affected positively if the pests being removed from within the fences are predators or competition, but also potentially negatively when the fence poses a barrier to the animal's free movement and therefore the gene flow. And that's what I'll be talking about today. So as part of a broader project, we set out to find a species that might experience both the positive and the negative effects. And in this particular part of the project, we wanted to find a species that likely moved between areas that are now separated by exclusion fences. And there are a few candidates, but the yellow-footed rock wallaby was the clear winner, and he's in light green here. I hope it's light green, I'm colourblind, I can't tell. The yellow-footed rock wallaby was ideal for this study because it lives in rocky outcrops that look like this. And these places, they like these places because they have these fragmented cliffs that make little valleys and holes and mazes that provide refuge from predators. And separating these colonies areas of very low habitat complexity, big open spaces that the yellow-footed rock wallaby would have to get between to form a metapopulation. And there are a few places where these open spaces are now also cut by exclusion fences, posing a further barrier. So to check it out to see how related these colonies were on either side of the fence, we had to catch them. And we also thought we'd see how often rock wallabies move between colonies in a connected habitat situation. So we also saw samples from the Gawler Ranges and the Flinders Ranges in South Australia. And you can see here that landscape down in South Australia is very different to in Queensland. Connected ranges of hills and cliffs and valleys rather than isolated mesas like I showed you in the last slide. And this here is us trapping in the Flinders Ranges at Nantawarra and IPA. So here's a map of the locations. We caught rock wallabies at 4 locations in the Gawler Ranges, and those were Organ Pipes, Yandinga, Stone Dam and Coolgundaby. 1 location in the Flinders Ranges at Wakawoodna Gap, and 2 locations in the Grey Range, where these rock wallabies are just out on little mesas separated by open flatness, and now also further separated by an exclusion fence down the middle. I'll rapid fire through these methods a bit because they're a bit jargon heavy, genetics jargon, but feel free to ask questions about it. We had 94 samples of yellow-footed rock wallaby and I extracted the DNA in the lab and sent that off to DART, and they returned to me 17,000 SNPs. And after filtering and whatnot, I had about 12,000 variable sites to work with. And from there, I performed a bunch of analyses, both in R and in other software programs. And I started by performing a typical population genetics approach to see how the populations were genetically structured. And you can see here that the Gawler Ranges are not the Flinders Ranges, are not the Grey Range, but within the Gawler Ranges, you can see structuring with Coolgundaby up here, Organ Pipes and Yandinga, which are geographically close. Geographically close. Come down here and Stone Dam out here. Ray Station here and Alaric Station structuring, and that was supported by the structure analysis, which are these bar graphs down the bottom. And why this is interesting is it suggests that the colonies are somewhat separate and that gene flow between them is irregular. enough for the colonies to be a little bit distinct genetically. So then we wanted to see if there had been any contemporary movement between these colonies. And to do that, because of the power of the SNP data set, we were able to perform a pairwise kinship analysis. And again, I'll spare you the long and the short of it, but basically it's the same analysis they've been using to catch the bad guys in the US. They run the DNA from the crime scene against the Ancestry.com database, and they find out that the murderer has a cousin who's a quarter Italian, and then they find out that she has a sketchy cousin, murder solved. But basically, this analysis generates a number between 0 and 1, 0 being totally unrelated, and 1 being an identical twin or a clone. And it reports this for all possible pairs. And in the rock wallaby context, this is what we found. This graph was produced by Gephi, which is a great program that does lots of things, but here it shows nodes, in this case, individual rock wallabies at their corresponding colony locations. And it connects the nodes with edges, which are these lines, and the thicker the line, the stronger the relationship between the individuals. So obviously everyone's a little related. So these I filtered these relationships to those only greater than 0.06, which are these thin lines running all over. And these relationships, in the case of the rock wallabies, go up to 0.05, which is a full sibling relationship, but here it's more likely a parent-offspring relationship. And you can see there's lots of relationships between the colonies in the Gawler Ranges, suggesting that in this connected landscape, there is regular movement between these colonies, which makes sense given the connectedness of the habitat. But most interestingly, here you can see a few first cousin relationships between Ray Station and Alaric, which is interesting because it means that the yellow-footed rock wallaby has moved this 8-kilometre distance across the open landscape within the last 2 generations, a movement that now won't be possible because of the placement of this fence right here. So from here, we decided to go one step further and do a first generation migrant analysis, which looks for the actual disperser. And the program, which is called Gene Class 2, identified 17 likely first generation migrants. And unsurprisingly, 15 of those were in that connected habitat. But interestingly, about a few of them had moved over 13 kilometres from this Yandinga population out to Kulgundabi, which is quite a distance. But most interestingly, two of the movements were this 8-kilometre distance between Alaric and Ray across that landscape, open and empty. So what does this mean for the rock wallabies? Well, most certainly in Queensland, it means that the movement behaviours between colonies now separated by fences will be disrupted. These are some genetic metrics down the bottom that may not mean a whole lot to a whole lot of people, but the basic gist is that several of these colonies are already showing genetic signatures of inbreeding and are quite small. And the implication is that these isolated colonies might need these inter-colony movements to stop them from sinking. The management implication is that in Queensland, there may need to be augmentation, particularly where single colonies or patched habitats are isolated by fencing. These results also have some implications in South Australia. They weren't just a connected habitat reference. Typically, foxes and goats are controlled around core colonies in South Australia, but these results show that broader landscape level management of these pests might be needed to allow for typical metapopulation dynamics, such as the wave and tide movements into and out of suitable habitat in response to resource availability. And also the spread of rock wallabies to areas of suitable habitat for the establishment of new colonies. Yeah, so that's pretty much it as far as the case study of yellow-footed rock wallabies goes, but I just wanted to talk quickly about why this approach is applicable in a broader wildlife management setting. This approach would be interesting to anyone that's interested in contemporary movement of individuals between populations, whether that's pigs up and down valleys, deer between ranges, fish between streams. The non-genetic approaches to answering the same question, things like GPS tracking or capture mark recapture, which rely on the individual you've collared or marked making the movement during the study period. If none move, or some do, just not the ones you've marked, you'll miss the movement entirely. With this kinship estimate approach, as long as the individual sampled are somehow related, you'll be able to detect movement regardless of when it happened. I've also had it said to me that there was no point going to the trouble of a kinship analysis like this because typical PopGen approaches, population genetics approaches, such as genetic structure analysis and gene flow will do the same thing. But that's actually incorrect. Those analysis show indicators of movement of genetic material between populations, not individuals. But genetic material doesn't have to go from population 1 to population 2 in a straight line. Genetic material can flow between multiple generations and multiple unknown populations. But if you wanted to detect that direct movement, kinship analysis and population assignment is the way to go. And that's pretty much it. So thank you. I particularly want to thank Holsworth Wildlife Research Endowment, who funded this research, and also the Bunthamara, Atnamatna and Gawler Ranges Aboriginal people, whose traditional lands these samples were collected from. Thank you very much. Any questions?
Thank you very much, Neem. That was very interesting. Nice videos, chasing the animals along. Any questions? To everybody who's joined us in this session, questions can be put into the Q&A box or into the